Okay, now we'll talk about chromatography and separating substances, which is topic 3.9, separation of solutions and mixtures, chromatography. Components of a liquid solution cannot be separated by filtration, unlike a colloid. So last time, in 11.5, we discussed how colloids can be separated through filtration, for example, milk or dust in the air. But a solution, a true solution, cannot. For example, if you have salt water, that's a solution. Salt cannot be filtered out of salt water by filtration. It will just go through the filter. And so we can use a chromatography. And this has a mobile phase and a stationary phase. The mixture, which moves on the stationary phase, is the mobile phase. So here, and we'll do a lab right at the start of Unit 3, showing a chromatography, and you probably had one or may had one in general chemistry as well. The liquid is the mobile phase, and it will move up the paper or the TLC, and then it takes the pigment with it. And the stationary phase is the paper, because of course the liquid from the bottom moves up, so it's mobile, and the stationary phase does not move because it's paper, so it's stationary. Solutes more strongly attracted to the mobile phase will move more in the mobile phase. So remember, like dissolves like. So if the mobile phase is polar, if we have a polar mobile phase, then the solutes which move in it the most strongly or the most will be polar. If we have a non-polar mobile phase, it would be the opposite, right? Because like dissolves like. Solutes more strongly attracted to the mobile phase will move more in the mobile phase. So polar attracts polar, non-polar attracts non-polar. Explain what is shown in this image. Let's say the solvent was water, which is obviously polar and can hydrogen bond. Describe what types of mobile phases would have moved farther or less far. I shouldn't use mobile phase. Describes, because the mobile phase is the water. Describe what types of inks, let's say, would have moved farther or less far, because the mobile phase is the water, the stationary phase is the paper. Go ahead and answer this, and then we will go over it. So you should say these inks would move the farthest, and that means that they are the most polar and or can hydrogen bond, and so interact the most with water. So the solvent is the mobile phase, the water, and the paper is the stationary phase. The colors which move the furthest would be the most polar and or can hydrogen bond. The colors which didn't move or hardly moved are likely very nonpolar. Instead of paper, we can use the same thing with thin layer chromatography, also called TLC. It's like basically fancy paper that does the same thing. So if in AP Chem, or if a question references TLC or thin layer chromatography, it's a chromatography like the one we just talked about. There's also column chromatography, which is very similar, except the stationary phase is a silica gel in a glass column, and then the solvent moves through along with the inks. So the solvent gets poured through the gel, and then depending on the inks and how much they're attracted to the solvent poured through as opposed to the gel, that determines how fast they move. So like, for example, in this case, if the solvent being poured is polar, and let's say that the gel, the stationary phase, is nonpolar, then what would that tell us about the polarities of the yellow and blue thing? Pause to think about it if you need to. Okay, hopefully you have an answer and you paused and you thought about it. So if the gel, the stationary phase, is nonpolar and the solvent being poured in is polar, then that tells us that the yellow ink is more polar than the blue, which is less polar here. Another type of separation we have is distillation. Distillation, as the standards for AP Chem say, separates chemical species by taking advantage of the differential strength of intermolecular attractions between them and the effects these interactions have on the vapor pressures. In other words, different substances have different boiling points and vapor pressures, and so they will boil at different times, and therefore you can separate them based on their difference in boiling point. So this is what distillation looks like. Unfortunately, we don't have the tools to do this one. You need a little bit more than what you have in most general chemistry classes. It would be fun, but we can't quite do it. But you can see that you heat water, and then you collect it, and there's a tube which has water on it, and then one of the liquid, there will be like two liquids in here or two substances in here, and one will boil faster or at a lower temperature than the other to separate them out. So this is another image showing the same thing. So in this case, if you had salt water, then the water boils and gets collected. So the water boils, it comes here. It's cooler in this tube, and so then it becomes liquid again and drops down into here. Whereas the salt, of course, does not boil, and so it remains behind as pure salt. So distillation generally involves three main steps: conversion of the desired liquid from a mixture into a vapor, the condensation of the purified liquid, so in this case pure water, and then the collection of the condensed liquid. So you wouldn't burn all of the water, or you wouldn't heat all of the water in this case, because then it would break your glassware. So you would leave just a little bit with all the salt left in there and probably dump that. And so you should be able to explain, for example, like what I just said, how salt water, the salt and water could be mostly separated using distillation. And understand that the salt is not going to boil until a much, much higher temperature, and so the water can be collected separately. And similarly, you could have something like this, ethanol and water, which could be separated using distillation. And it's the same thing. Depending on the boiling points, the lower boiling point thing is going to be boiled off at a lower boiling point than the higher boiling point thing. So that's why I say here, the temperature of the solution must be kept at the boiling point of ethanol, or slightly above it, but significantly below the boiling point of water, because ethanol has a lower boiling point. So in something like this, the ethanol is being boiled off, but it's too cool for most of the water to become gas. And so the ethanol gets collected on the other side, and the water stays. Okay, lastly, I just want to take like 2 minutes to talk about this last slide. So topic 3.11, I honestly feel like it hardly even comes up. Maybe there's a couple unit 3. Rose, I'm finishing up here, okay? Maybe there's a couple unit 3 MCQ questions, but it's not a real focus point because there's not a whole lot you can extrapolate from it, I don't think. But understand this. You basically have to memorize microwave radiation is associated with rotating molecules. Infrared radiation is associated with vibrating molecules. And UV is associated with transitions in electronic energy levels. So again, microwave, molecular rotation, infrared, vibration, and then UV or visible is associated with electronic energy level changes. How would I remember this? Well, I think there's a couple things. One, you can remember in general chemistry that we do a lab where we heat chemicals and they release light. And if you remember the explanation from general chemistry, that this is the same kind of thing used in fireworks, and that it's differences in electronic energy levels. So if you can remember that, and remember that that was visible light slash UV light, well then that's one that you already have from general chemistry. The other thing I think you can just look at is, if you look at their wavelengths, you can see, okay, UV and visible light are the shortest wavelength. That means it's high energy. And then infrared is like the middle energy. And then we have microwave, which is like the lowest energy because it's longer wavelength. So that means it's lower energy. And in topic 3.12, we'll go over this just a little bit more. So how does that help? Well, if we can remember that, we can think, okay, it kind of makes sense that lower energy, the lowest energy, is just going to, microwave is just going to rotate the molecule, okay? Just take some of those bonds. You know, you have a bond here. There's atoms. I should have got a molecule out, but I didn't. And you just rotate, right? Maybe you have three groups attached to a carbon, and just their position gets twisted. Okay, so it'd just be like if you have one of those models and you're just twisting around the axis. So let me just doodle it here so there's something you can see. Right? If we're just twisting the molecule, rotating, you can kind of see how that would be relatively little energy to be able to do that. So that's microwave. Then in middle energy, vibrating, if we're causing the atoms to move a little bit more, right? Maybe they're getting closer, further away from each other, something like that. Okay, you could see why that would take a little bit more energy, right? And then, okay, highest energy, UV slash visible light. Well, you can remember the lab you did in general chemistry where visible light shone off. You can also think UV, visible light, higher energy, enough energy to move electrons. So that's all you need to know from 3.11, and there's not really much more to say about it than that. You don't need to know a bit more in depth than that, so I just think you just have to memorize those 3 points, but there's just a little bit of logic there, and hopefully my logic on that is correct, by the way, because 3.11, I honestly don't know that much about this particular thing. Just throwing that out there, but just know those 3 points. Okay, microwave is rotational, and then infrared is vibrational, visible slash UV is electron energy level changes. So I'm just throwing that in to talking about separations because there's not that much to talk about. Oh, and also know that sometimes they might mention evaporation is another technique. So I didn't talk about in this video, but you could use evaporation to separate, for example, salt from a liquid. Just leave it out until the water evaporates, and or use desiccators, which we've already done in class, so you should be familiar with that. Okay, that is it for chromatography and other separation techniques to separate substances.